So I've worked at cell signaling technology for 10 years, and I want you to treat me today as a resource in how antibodies perform across protocols, because I've tested over 1 million independent antibody samples using various protocols and immunofluorescence cytometry. So I have a wealth of knowledge on how these perform in different assays, different protocol types. Okay, everyone's pretty familiar with CST, it looks like. All I'll say then is that I came to CST having worked 6 years in academia, and making that jump into the corporate world, I was initially pretty concerned about leaving that science behind. And what I've really appreciated working there is that there's a very academic focus at CST, and we bring in leading scientists every week for seminars within the company, and we get to interact a lot with scientists, both at conferences like this and in collaborations, but most importantly through technical support. So when you're using antibodies and you have questions, if you're using flow cytometry and you submit a technical support request, that comes to my group, and we get to see the problems that you're all having. We answer the questions, we help you solve those problems, get things working. As a result, we see what those common problems are, and that's where this workshop grew out of, is seeing these problems that arise and wanting to answer some general questions that people have regarding detecting phosphorylated proteins, and more generally, just performing intracellular flow cytometry, detecting anything inside the cell. So this is a general list of topics, and I've structured them around those questions that we receive. So a lot of you are doing detection of phosphorylated proteins already, so you kind of have the answer to this question already. This one, by the way, was not a technical support question. I wrote this one because it was a great way to lead in. So when you think about what you're usually doing by flow, a lot of people do is live cell flow, and you've got access to 370 CD markers and about 1,400 proteins on the plasma membrane surface that you could detect with antibodies in a live cell assay. Of course, if you fix and permeabilize those cells, get into the cell, now you're talking about an order of magnitude more proteins that you can detect, and over 400,000 modifications on those proteins as measured by mass spectrometry. And that's really where the biology of these cells is driven. These are the readouts that let you understand what's going on in the cell. What is the cell doing? Where is the cell headed in terms of differentiation or activity? And we're just scratching the surface of what these marks mean. So this is a very exciting time to be involved in reading out these phosphorylation events within, especially the immune cell component, because we're just starting to understand what these modifications do in immune cells. So phosphorylated proteins, these phosphate groups get attached to proteins, and this controls the function of these proteins in many cases, and those make up the majority of these post-translational modifications, or PTMs. And traditionally, that's been detected by a Western blot, and many of you have, show of hands, that had done that before. So you know that Western blot is great at this. It's a very nice assay for reading out phosphorylation. It's got advantages like a visible protein size. So you can see if the antibody you're adding is detecting the protein you think it is. If it shows up at the wrong molecular weight, then you know that this antibody is not working. And you can also see if it's sticking to something else. And you can decide to just ignore non-specific staining if it's at the wrong size. And these are two things that you can't do in flow. If you're getting fluorescence coming out of a cell, you're trusting that antibody is sticking to what you think it is. And you're also trusting that all that fluorescence is coming from the thing that you think it is. So Western blot does have the advantage of being able to give some orthogonal validation that your antibody is doing what you think it is. It also kind of standardizes the landscape for that antibody. Since you're lysing these cells, you're denaturing the proteins, you don't need to worry that some kind of treatment that you're doing has caused a translocation, and now the antibody is not going to bind to your phosphorylation site because it's in an organelle that your protocol isn't enabling the antibody to access. It's busting everything open and linearizing it, and the antibodies have a grand old time. But the disadvantages, of course, are that because you're lysing all of these cells, you can no longer tell what cell populations the signal might have come from. You can't multiplex as easily as you can by flow, and of course, and it's slower. So going back to live cell flow then, again, the antibodies can't get into the cell. Antibodies towards CD markers, it's fantastic. You mix them, you add them to the cells, piece of cake, throw it in the cytometer. But an intracellular target, an antibody trying to get to that intracellular target, can't get into those cells. And this is where protocols abound. And this is what we're going to try to help today, is discussing what these differences mean within your protocols and how they can affect the antibodies that you're using. So that leads right to the first question. What protocol should I use for Phospho-flow? And this is a question we get quite a bit. And there's been, due to some early publications that show that methanol permeabilization was the best way to get phospho signal in Phospho-flow. There's this feeling that there's just one protocol that is going to be required for detection of phosphorylated proteins. And I'm happy to tell you that that's not the case. You have a lot of flexibility. However, that also can mean it can be scary because there's many different options. So a variety of protocols will work, but in general, I want to just run through a quick overview of what the protocol looks like in general. So you're taking your cells, and in order for those antibodies to get in again, you have to fix them, you have to permeabilize them, you're adding antibodies, you're incubating, you're washing out the antibody, and then reading on your cytometer. And the things that are going to affect the specificity of the antibodies that you're using include how you fix the cells, what reagents you choose, how long you fix for, permeabilization reagent that you use, concentration of the antibody you're adding, the buffers that you're incubating them with, and the buffers that you're washing with. And I'm going to focus most of the talk on the permeabilization reagents because these are the biggest difference that you'll see from protocol to protocol. And to give you a brief overview of broadly what these do, we can divide these up into selective detergents, which selectively permeabilize parts of the cell, non-selective detergents, which are going to permeabilize membranes throughout the cell, and then organic solvents. And to show you some examples of these and pros and cons, we've got a live cell here just for to kind of orient you. I forgot to explain what this is. Our little green nucleus here, blue cytoplasm, that's our cell. Selective detergents like saponin. Saponin interacts with cholesterol on the membrane of the cell, the plasma membrane. So you're enabling antibodies to now get into the cytoplasm, but it doesn't permeabilize the nucleus, so you don't have access to any nuclear targets with saponin. Non-selective detergents like Triton or Tween, these permeabilize membranes throughout the cell. So this is going to enable antibodies to get into the nucleus. But because it's more like you're adding Dawn dish soap almost. You're adding this soapy stuff that's disrupting these membranes. So you're going to lose some soluble membrane proteins if they're not buckled down. And then organic solvents like methanol are very thorough. They permeabilize things like lysosomes that even the detergents can sometimes have trouble with. But they're very disruptive to that protein landscape. So they change what that antibody is going to be seeing and can then affect the binding of that antibody. And I'm going to emphasize again that the cells need to be fixed prior to permeabilizing them, because if you add a bunch of detergent to live cells, you're just releasing enzymes and all their contents out into the wilderness, and you're going to cause all kinds of problems for yourself. Organic solvents like methanol can be used for fixation as well. In general, we focus on formaldehyde fixation for the rest of the talk. That's what I'll be centered around, is basically the assumption that everything is formaldehyde fixed. I'd be happy to speak with you after the talk if you have questions about methanol fixation. So I mentioned that protocol is flexible, and I wanted to just show some data to highlight this. So here's data showing phospho-S6. In blue, we've got untreated cells. These were Jurkat cells. I neglected to put that on there. Red is inhibited with a PI3K inhibitor. You can see that S6 phosphorylation is detected. You see a nice shift between these two. After formaldehyde fixation, whether you're detecting with formaldehyde fixation followed by methanol permeabilization, or if you permeabilize with Triton or permeabilize with saponin, you get very consistent signal. But other targets like the phosphostats in general require methanol. And even though they're in the nucleus, there are plenty of other nuclear targets that work fine with Triton. Something about the stats, you need methanol to go in there, and whether it's breaking up a protein-protein interaction or enabling access to this phosphorylation site by denaturing the protein slightly, we're not sure. But the point is that these differences in the reliance on these different permeabilization agents is going to be kind of driven by the antibody and the target, not the fact that it's a phosphoepitope specifically. So to answer that question then, to go back to what protocol you want to use, the best answer is to get as much information as you can from antibody manufacturers like ourselves. We try to make sure we provide that protocol that we know the antibody works in, well-validated publications. So if somebody shows this works, they've shown in the publication that they validated the antibody, and they provide the methods in the publication. And then if this has been done in the lab before, if you know the protocol in the lab, that's great too. And those working protocols are a good place to start because, of course, doing it with one product, one antibody is fine. It's building panels that becomes a challenge. To show more data, just emphasizing this isn't just phosphorylation, this is other modification types as well. So protein cleavage, protein methylation, protein acetylation. The data I'm showing here, all of the flow data we acquired with formaldehyde fixed cells, methanol permeabilization. All of the immunofluorescence images, formaldehyde fixation with triton permeabilization. And the antibody performs equally well in both. So to recap, look for protocols and protocol information as much as you can from known sources and follow the protocols, at least to get you going and understand how that product, how that antibody is going to work in your hands. But understand that your antibody could be flexible too. So you've got options for that, for those panels, and be able to build a panel around a more restrictive antibody, something that needs a protocol, one protocol specifically. And you need to permeabilize the cells, but there isn't one specific reagent that you need to use. So that leads to the next question then. All right, if protocols can be flexible, why isn't this antibody working in my protocol? There are a variety of reasons, but I'm going to focus first on really what happens with these protocol changes, and just to get you thinking about, from that antibody's perspective, what does it mean? So your different protocols can affect binding in three ways. It's going to affect the epitope, the landscape, and the antibody. So I'm going to show some examples for this. So it's going to affect the epitope. So here we've got an antibody detecting this green epitope on protein X here. In protocol 1, that's accessible. Protocol 2, it's not. This is a conformation change. This could be a difference between having methanol present, having a detergent present. It can also be, as I mentioned, a translocation, or if you've got a protein, say, on a lysosomal membrane where the epitope is now inside the lysosome. If one protocol doesn't allow the antibody to get in there and access it, then you're not going to get any signal. The reason I've got this red epitope there is to show you that this is not a protein-specific problem. You can have a different clone that could bind to a different epitope on that protein that might work in both protocols. Now, this is a little different from phospho detection, right? Where a phosphorylation site is going to be restricted to one epitope. But if you're detecting an intracellular readout, you're detecting protein in that cell, there might be other options, other clones that might give you flexibility to use different protocols. Okay, what about the landscape then? Think about when the antibody's in there are billions of other things that an antibody could bind to besides the protein that you want to detect. So we see this a lot where you've got one protocol, the antibody binds specifically, another protocol, whether it's exposing a different epitope or hiding one, and now the antibody has affinity, low enough affinity that it's able to bind to a different protein, you can get non-specific binding, and this can be affected by the reagents that you're using. And hand in hand with that is the antibody itself, because showing the same example now under the antibody group here, you could have a different clone binding the same site that might have higher affinity and won't bind to this non-specific purple protein down here that we're not trying to detect. So it's a function of where the antibody needs to bind. So can the antibody bind the epitope it needs to? The landscape, can it bind to something else if it's a somewhat promiscuous antibody, if it's not highly specific? And then the antibody itself. So ensure that the antibody is validated in the protocol that you're using in their lab. And we understand there's a variety of protocols out there. I'm not knocking them, but we just want to emphasize that it's not a guarantee that you can take an antibody and just because it's flow validated or any other application, whatever you're doing, doesn't mean that it's going to work in that application no matter what you do in your lab. So it's important to make sure that you're following the established protocol. I've got an example here for a keratin 5 antibody, triton, methanol. If you were reading this out by flow, you wouldn't know, these are MCF10A cells. They all are expressing keratin 5. But if you were using a different protocol and you were trying to, say, gate on these cells, you wouldn't realize that you're gating on just the subpopulation that's not actually representing the full expression of this protein. even more so to just take a step back to the Western front again. If you're moving into flow from Western, it's important that you use flow-validated antibodies, things that are known to work in flow, because there are plenty of Western antibodies that work in Western blot that just don't make the leap into assays when you've got native folded protein. And this is an example from one of our development projects at CST where we had antibodies, 4 different clones here to heat shock protein 70 that we were developing. By Western, they look fairly similar. By immunofluorescence imaging, though, you can see they give completely different staining patterns. So if you're running these cells through a cytometer instead of looking at them on the scope, you would never know that the fluorescence you're getting isn't heat shock protein 70, which should actually look like this. Okay, so then when you're validating these antibodies, another reason that or when you're testing them, if you say, this isn't working in my assay, understand that phosphorylation events are temporal. So unlike detecting total protein levels, this is going to change through time, and some events can be degraded very quickly after that cascade is complete. So what I'm showing here are data for PBMCs treated with anti-CD3, CD28 for the times that I'm indicating here. You can see that SLP76, which is an indication of T-cell activation, is up within 2 minutes, and by 30 minutes is already decreasing. And S6, which is phospho-6 is an indicator of cell proliferation. This site is phosphorylated when cells are growing and proliferating. Again, comes on very quickly and is gone. So we get questions, hey, I activated my T cells for 7 days and I don't see any phospho-76 signal. Yeah, you're 7 days too late. So be aware that some of this isn't a permanent mark. These things come and go on the proteins very rapidly. So understanding the biology of your system is important. And lastly, I want to say about fix-perm kits. These are widely used. That's fantastic. They're very convenient. Just make sure, again, that your antibody is performing in those kits. Examples here, again, fix-and-perm has shown 2 different commercial kits. This is total AKT. This is not phospho-AKT. These are jerkit cells. They should all have a lot of AKT in there. But you start getting some differential signal with these 2 kits. At the same time, we ran, again, phospho-S6. We don't see that this double peak or anything there, but then gamma H2AX or phospho-H2AX, you do again see this signal. So, this kind of double staining. So again, it's just something important to be aware of, that you want to know if you're shifting protocols, or if you're taking something that was known to work in one protocol and you're like, oh, you know what, I'm just going to switch, I'm going to start buying this kit, it's more convenient. Kits can be tough because you don't always know what's inside them. So make sure that it's working the way you think it is by doing some validation work on your end as well. So, why isn't it working? Make sure you're using a flow-validated antibody. Understand that changes in your protocol are going to affect that performance, potentially. And for phosphorylation sites, if known, then leverage that knowledge about the biology of your site to know timing-wise when that should be up, so you can detect it at that maximum phosphorylation. Okay, so, of course, again, doing one antibody by flow, that's not necessarily the hard thing. It's now, I want to phenotype, I want to add these into a panel. And this is another big struggle that people have. And so we wanted to be able to just show a brief overview of some options that you have to get CD markers working in your assay. So understand that within that general protocol I showed of fixing and permeabilizing and antibody addition, you can add CD markers at different times within those steps. So the simplest thing for you to do as a researcher would be to take your sample, fix it, permeabilize it, and add everything all at once. Your CD marker antibodies, your phospho-specific antibodies, any intracellular antibodies, throw them together as a cocktail. You only have to incubate once, wash once. That's great, but depending on the permeabilization you're using, those CD markers may not bind, the antibodies may not bind those CD markers as well as they would in live cells. So another option is then to take your CD marker antibodies and add them to your live cells. Then formaldehyde fixation is going to cross-link those CD marker antibodies onto the proteins on the surface. You can permeabilize and then come in with your phospho-specific antibodies at the end. Gives you much better differentiation for your CD markers, But it does make a little extra, gives you some extra steps that you have to use in the lab. And solvents, if you're using methanol, can damage some fluorophores. I'll show you data for that in a second. So then a third option is to fix the cells first, stain with your surface markers, permeabilize, and then stain with your phosphos. Why would you want to do this? What's the difference? Well, just understand that with live cells, if you're adding antibodies, you're adding things that are binding to those cells, you can affect signaling within those cells. It's something to be cognizant of. Also, it's going to take some time. So if you've got a short activation that you want to perform, you might want to perform that treatment, fix the cells, get everything locked down so your timing is controlled, and then come in with your surface markers. So to show you a bit of data to emphasize what I was saying, so this is that first option I was showing, adding everything at the end. And I'm showing two different clones here, one for CD3, one for CD4. How they separate on live cells, then if you formaldehyde fix, permeabilize with saponin, and then and then you're adding the CD markers afterwards. But then here you can see now if you permeabilize with methanol first, your CD3 signals now dropped quite a bit. We can still tell the two populations apart, but there's less differentiation between them. And Triton, interestingly, you don't lose signal for your positives, but you gain signal in your negatives. So now Triton is doing something that's allowing this CD3 clone to bind to something non-specifically, as we were discussing before. So it's up to you as a researcher to decide, you know what, I can live with this. I can still differentiate my positives and negatives. I'm going to go with this protocol anyway. But it's something to be aware of. And you can see CD4 was pretty consistent among all the protocols. Okay, so now what about adding them first? What can happen here is that, again, things like methanol can affect, if you've got protein fluorophores like PE or APC, methanol can actually denature those fluorophores and make it so they don't work anymore. So here's CD3 conjugated to FITC. This was added live, and then following these different permeabilization steps, you don't really see a change in that fluorescence. But PE, you lose your positive population completely once you've permeabilized with methanol because it's denaturing that big PE protein and it's no longer fluorescent. So to summarize, you can do this. You can get phenotyping and phosphorylation in the same assay. It's just helpful for you to understand what's the optimal timing for you to use, what fluorophores should you use to avoid any bleaching if you've got to use any harsh solvents. And then what I'd recommend is if you have CD markers that you use frequently in your labs, you might have to do some work up front, take your cells through different protocols and understand how that affects the staining that you're getting from these different CD marker antibodies, but then write it all down. And that way downstream, if you're going along, you're doing this research project, and you have to detect phospho X and phospho Y, and you're months and months later, you know what options you have. And so if this one antibody, all of a sudden you realize, I have to use a different protocol than what we've been doing all along. But I know that I can use these 3, 4, 5 CD markers in that protocol, then that's fantastic knowledge to have. All right, so the other question we get frequently is, following up on this didn't work in my protocol, can you show me some extra data in a model system similar to what I'm doing, just so I can see, can you show me that this works, just to give me something to hope for, right? So what I want to do is just show a simple 6-color experiment doing a T-cell activation assay. So doing the same CD3, CD28 stimulation, a 10-minute stim. It's just to give you an example of what I thought about going into this. I was thinking, okay, this is a short treatment time. The way we do this, we have soluble anti-CD3, soluble anti-CD28. We add that to the cells. We cross-link with a mouse, anti-mouse IG, cross-link those on the surface. So it's a cross-linked, and then you're throwing them back at 37 degrees. You've done this all on ice, so the cells are kind of on hold. Putting it back at 37 degrees for 10 minutes. So it's a short treatment time, and you're adding antibodies to the surface. So I didn't want to have my CD markers on the surface of these cells, potentially interfering with the binding of this CD3 treatment, and also potentially causing signaling cascades downstream in the cells. I wanted to make sure I eliminated any dead cells. We were just thawing, this was a frozen vial of PBMCs, so we want to make sure we get rid of cells that were dead upon thawing and that wouldn't be responding to the treatment anyway. And I knew I'd be permeabilizing with methanol for the readouts I wanted. So I knew I wanted to avoid using PE if I was going to add antibodies first. So what I chose to do is use a ghost dye. This is a fixable viability dye. These are great if you haven't used these before. You add them to the live cells. They are fixed and they stay within the cells so that you know what was live and what was dead at the time of fixation. I was phenotyping with CD3, CD4, and CD8, and then my signaling readouts were phospho-76 and phospho-S6 again. So I decided to do option C here, where I was fixing the cells first. I wanted to lock in that 10-minute time point, come in with my CD markers, permeabilize, and then come in with my SLP76 and S6. And the reason for that is because CD8, I didn't show the data before, but CD8 does lose some signal if you add it at the end of all of these steps. So how does it look? Well, first of all, so here's our PBMC population. This ghost dye, this fixable viability dye, it gets brighter in dead cells, similar to how propydium iodide works. It can't cross the membrane of live cells, so you just get a little bit of staining in live cells, and if the cell was dead, then it can cross the membrane and stain things intracellularly. So here's all of our live cells. We gated on those first. Gated on our CD3 positives. And from there, took our two CD8 and CD4 positive populations. And the reason I was doing this experiment, by the way, was I was just curious, is there differences in SLP76 phosphorylation between CD8s and CD4s when you activate these T cells? I hadn't seen anything about it, and I was wondering what we would find. Sadly, for the purposes of this presentation, there wasn't much difference. There was activation in both populations, and it looked very similar, actually. So what I'm showing here in dark, in the solid color, I should say, for both graphs, is the treated population. The lines through it is our untreated population. So you can see SLP76 phosphorylation coming on in both of those treated populations, and then S6 coming on equally in those treated populations, but very similarly, actually, with the CD4s and CD8 positive T cells. So, with that, the summary overall. Oh, yes, yeah.
Can you just say again what order the steps were that you did this 6-color?
Sure. So the question was, can I say again what order I did this 6-color staining in? Yeah, we've got time, actually. I can give you... So the ghost dye I didn't mention before, and that doesn't fit into the protocol I was showing before. That I added ahead of time. We had done some experiments before to show that we couldn't see any differences in any signaling nodes that we checked by adding that onto live cells. So I added that first. And this is all while the cells are on ice. Came in with anti-CD3, CD28, added those, let them incubate for 15 minutes, washed it off. Came with anti-mouse IG to cross-link those, again for 15 minutes, washed that off, and then fixed formaldehyde, added the CD markers, permeabilized with methanol, and then added the phospho readouts. I'm sorry? Oh, so the question, APC is not sensitive to methanol. Yeah, that's a good point. So, sorry, fruit fly. So APC, it does lose some signal. I took the data out of this talk because I knew it'd probably run long. It loses about 50% of the signal, but you still get the same percentage of positive cells, but you just lose some intensity. So the overall summaries, and I'll take questions in just a minute, but overall, respect the protocols. If you've got a published protocol or a protocol from a manufacturer, follow that religiously at first. Because even swapping things out, like, oh, well, in our lab we wash with PBS with tween instead of just PBS. We just don't know how this is going to impact binding. But know that there are many antibodies that are going to be flexible, and these are giving you options to be able to use these across panel builds, or if you get, if you're, say, doing tissue preparation, you're required to prep this tissue in a certain way. Well, that doesn't mean the antibody is just flat out not going to work. It just means you should do some testing to make sure. You can do phenotyping along with phosphorylation readouts. And more importantly, again, technical support comes right to my team. Everyone who's doing the flow validation at CST are the same people who are going to handle any tech support questions that you have. So you can go to our website. You can just mention that you're doing Phospho-flow. There's a drop-down that asks what application you're doing. And that's going to be coming right to the people who are doing all the validation work at CST. So we'll be able to help you along with that assay. So thank you very much, and I'll take any questions.